Our first speaker is Noel Lowndes, who's Professor of Biochemistry and Director of the Centre of Chromosome Biology here in NUIG. Noel graduated with a degree in genetics from Trinity College Dublin in 1985. His postgraduate studies were completed in Glasgow and Columbia and in New York, and he was awarded his PhD in 1987 from the University of Glasgow. He has conducted university-level research in the National Institute for Medical Research in London. He established his own laboratory at the Cancer Research UK at Clare Hall Laboratories, and he is widely regarded as one of the world authorities in the field of DNA repair and cell cycle research. He returned to this university in 2001, and he took up the chair of biochemistry in NUIG. And since that time, the department has significantly developed its research capacity, in particular in this area. He is the founding director of the Centre for Chromosome Biology. It gives me great pleasure, therefore, to introduce Noel Lowndes.
Thank you for the generous introduction. I'd like to start by thanking Ashla and Jerry for the invitation to speak. In particular, I'd like to thank Ashla for being so persuasive, and our conversations together I've greatly enjoyed. She's been trying to point me in the right direction. So I'd like to start just with a gratuitous pretty picture. This is from one of my PhD students who's got a major interest in photography. And this is Galway under better weather conditions, and you should all come back and try and enjoy it. Okay, so briefly the outline of my presentation today will be start with genetics, history and key concepts, the genetic and epigenetic material, which is DNA, and of course the associated proteins. DNA does not exist in cells as naked DNA. It can't exist in cells. I'll explain why. It's always coated in proteins. Move on to some genetic testing and how phenotypes are revealed from this. I think the most important thing that's happened in genomics since Mendel discovered the field of genetics is in fact the human genome. And this will, as you're all aware, have major impacts on society. And then I'd like to finish with some brief discussion of some future perspectives and the major scientific challenges ahead, because there is rather a lot of hype about the Genome Project. Okay, so genetics, history and key concepts. This is a one-slide whiz through the field of genetics. And it starts with this guy, Gregor Mendel, a Czech monk, an ethnic German, who experimented with peas in the late 1800s, 1860s, and discovered, in fact, that traits are passed in discrete units, which later became termed genes. Up until this was crazy, wacky theories about hereditary. In Europe, it was favored that genetic traits were passed on in the father's sperm, and the mother had very little to do with it. She was just a receptacle for the father's sperm. Aristotle had even weirder ideas. I'm not even going to go into them. They're vaguely obscene. About the time of Gregor, it was generally assumed that the coming together of genetic traits was like mixing paint, like mixing two colors and getting a third color, that there was no discrete separate entity. So it was the Danish botanist Wilhelm Johansen that coined the term gene for the unit of inheritance. Neither of these studies had any clue about the physical basis of the gene, and that's one of the characteristics of genetics as a science. It is a very abstract science in its pure form. You can study genetics without even understanding what a gene is. And for 50, 60 years, there was a lot of geneticists, and they hadn't a clue what a gene was, but they were still able to make good careers as geneticists. Thomas Morgan, working on fruit flies, showed that genes sit on the chromosomes. The chromosomes have been visible down microscopes because they can be very large. They can be easily visible for since Leeuwenhoek 130 years or so ago. But no one knew that they were the sites of the genes. So he showed this in this fruit fly organism here. And this led to the idea that genes are arranged along the chromosomes like beads on a string. Fungal geneticists interested in metabolic enzymes came up with the concept because they were studying enzymatic pathways, that one gene equals one enzyme. And one gene, one protein led from that. And that's not the case. It's far more complicated than that. Now we get into the stage where we're beginning to get to grips with the physical nature of what a gene actually is. Oswald, Avery, Colin MacLeod, and Macklin McCarthy discovered that genes are made from DNA, a chemical that was known to exist in cells. No one knew what it was. Rival theories thought it was proteins, or the units of genes. But these guys were the first to show that genes are actually made of DNA, this famous double helix molecule. Then, of course, Watson and Crick actually discovered what the structure was. They used other people's data to build models to infer the structure, and published a very nice paper, a seminal paper in the scientific journal Nature, and won a Nobel Prize because of it, for their inspired guesswork, basically. Roberts and Sharp then showed that genes were split. They weren't just, each unit of hereditary isn't just a single unit, it's actually split. The gene is split into different parts, which means those different parts can be rearranged, which means one gene can give rise to more than one protein. And another type of genetic element, RNA elements that aren't even translated into proteins, were discovered in worms. C. elegans. This is the worm that they discovered these regulatory RNAs in. So prior to the sequencing the human genome, no one really knew how many genes there were. I mean, I did genetics at Trinity, graduated in '83, and I can remember being lectured on the number of genes in the human genome, and estimates ranged from 2 million to 1 million, to a couple of 100,000 to 100,000. And as we move on, 2 years ago it was 30,000, then it was 25,000, now it's 22,000. So the number of actual genes, depending on how you define them, is declining the whole time. The definition was protein-coding genes. Okay? However, more recently, with the sequencing of the genome and subsequent studies, the idea is that genes are part of a wide spectrum. There may, in fact, be hundreds of thousands of genes, depending on how you define a gene. We may be able to come back to that. Now, before discussing genetics, I think it really is important to focus on the father of genetics. What did Gregor Mendel do to establish this field, the whole field of Mendelian genetics? Well, it's quite simple. He experimented with peas in a small plot in his monastery, and he did things like this. He took a pea flower, he emasculated the pea flower by cutting off its male parts. As far as we know, plants don't feel pain, so this was okay. He then took pollen from another plant. This one had white flowers, as it happened, and fertilized the purple flowering plant. And then got the peas, planted them, and this is what happened. All the flowers were purple. So this is what's happening. If you cross a plant with purple flowers with one with white flowers, all the progeny are purple. Clearly purple is dominant, okay? Whereas whatever gene is controlling the white phenotype is recessive. And you can show, and what he then went on to show is by breeding these plants together, he could show that the white phenotype in the next generation then segregated in the 3-to-1 ratio. And this was extremely good evidence that genes were made up of individual units, and that those units could be either dominant or recessive. He didn't just look at purple and white plants. He looked at plants where the flowers appeared axially or terminally, where the peas were either yellow or green, round or wrinkled, inflated, constricted, green, yellow, etc. These are all the traits he looked at. And in all cases, this is the actual data, he got ratios of effectively 3-to-1. So traits must come in discrete units, which we now term genes. So what about Mendelian genetics in humans? Okay, well, here's some nice trivial examples. This is the widow's peak. This, when the hairline comes down into a peak. These are two sisters, and you can see that one sister has the widow's peak and the other doesn't. And this is, in fact, a dominant Mendelian trait, because when you study the pedigree analysis, you can see that 2 copies of the dominant gene, or even just 1 copy of the dominant gene, is enough to give widow's peak, whereas 1 in 4 of this generation will not have this. Another example, those of us who have attached earlobes, I do, as opposed to detached earlobes. Well, the attached earlobe is a recessive Mendelian trait. So we have got 2 mutations in the gene that determines this particular trait. So these are fairly trivial Mendelian traits. They don't have dire phenotypic consequences, cosmetic consequences only. But how about Other Mendelian traits, for example, the gene that causes cystic fibrosis, which is very prevalent in Northwest Europe. Well, this one does have very serious consequences. There are many, many examples of this, of Mendelian traits that impact on disease. Now, traits are not only inherited in Mendelian ways, but they're also inherited in non-Mendelian inheritance patterns. And these are always a lot more complex than predicted by that very simple Mendelian genetics that Mendel first showed with peas. They're polygenic traits. What that means is more than one gene is involved. Things like human height, a very good example. It has an environmental impact, but it is quite tightly controlled by the genes. So if you've got tall parents, the children will be tall. You've got short parents, the children will be short. There are always exceptions in science, but in general, it's very true. And what's happening here with these polygenic traits is they're determined by the sum of the individual genes, some of which will be negative. Take height again. Some genes could cause short height, some genes could control tall height. It's the sum of all those genes that determines your height. Diet does have an impact. The environment is always a factor. So even though height, for example, and skin color are polygenic traits, they're also multifactorial traits. Multifactorial traits are those that depend not just on the genotype, but also the environment. So in this case, it's the sum of the effects of all the genes, positive and negative for the trait, and the environmental factors that contribute to the phenotype. Okay. Now I just want to move on to the physical basis of genetics, and that's the DNA itself. And this is in fact the famous model that Jim Watson and Francis Crick built and published in 1953. They used other people's data. They didn't do an experiment of their own. They built a model. They cut out the shapes of the various chemicals that they knew were involved, components of DNA, and assembled them into this structure, which you can't see very well. But this is the original structure that they assembled to explain the data from other people and published in Nature. Here's a more modern view of DNA from a textbook. Now, what do we know about DNA is that it's composed of two strands. The genetic information is contained within the bases that are on the inside of the double helix, and they come in 4 flavors: T, A, C, or G. T always pairs with A, G always pairs with C. And the two strands are connected by these pink red dots. These represent chemical bonds, sort of hydrogen bonds. The important thing about chemical bonds is they can be hydrogen bonds, they can be broken, whereas these bonds here are very hard to break. So these easy to break bonds means you can rip the DNA apart. Once it's ripped apart, it now forms a template. If you use enzymes called DNA polymerases, they can recognize that T and put an A next to it. Recognize a G, put a C next to it. So Watson and Crick realized this, that the structure of this molecule very elegantly explained how DNA is replicated. Okay, so this is how you often see it in textbooks, but of course it's not, it's more like this, because atoms have a volume. And you can see at the outside of the molecule here, these yellow things here correspond to phosphates. So you've got phosphates on the outside, the blue is sugar, and the bases are kind of internal, okay? These phosphates have a negative charge, and that's important from the point of view of genome dynamics. But for the cell to interpret this structure, it somehow has the proteins that recognize DNA have to fit into these grooves. This is the major groove, this is the minor groove. And these proteins literally have feeling fingers that can feel along the DNA, and then when they find a fit, a sequence that they like, perhaps T, G, C, A, they lock on. And then they either turn on gene expression or turn off gene expression. Okay, so a very brief introduction to DNA. But as I mentioned at the beginning, DNA is not found in the cell as a naked DNA molecule. A naked DNA molecule, because of all those phosphates and the electrostatic repulsion that you would get, negative charge repels negative charge, just like trying to put the two north poles of a magnet together, exactly the same thing. The volume of the genetic material would far exceed the total volume of the cell. It's a huge random coil that wouldn't at all fit into the cell. So what the cell does is it wraps the DNA around proteins that have positive charge, and the positive negative charges neutralize. Now you can compact the DNA down into a very small volume indeed. For example, the diameter of DNA is 2 nanometers. 2 nanometers. That's 200 millionths of a meter. So one of the secrets behind compacting DNA into a short volume is, in fact, that it is incredibly thin. The amount of DNA in each one of the cells in your body is about that much. If you put it out end to end, about 2 meters. So you've got a 2 meter long molecule that is broken up into chromosomal sized bits, but 2 meters in total length, compacted into something that's 5 millionths of a meter in diameter. An extraordinary compaction problem, and it's achieved by these proteins that the DNA is wrapped around. They're called histones. That's not important. What is important is that they neutralize those negative charge. So this is the first level of compaction. The DNA is wrapped twice around these beads to give a diameter of about 11 nanometers. Then these beads are further. Everything's helices in biology. The DNA double helix. The DNA is then wrapped around these things to form another helix. And then this helix of helixes wrapped again to form a helix of helices, right? And this is a 30 nanometer fiber. And then you get even larger fibers where these helix of a helix of a helix is now coiled into even more helices. This is what a chromosome would look like. So there are proteins around the base here that attach the base of these loops. But the DNA in this sort of structure is looping out into these amazing loops. When it comes to segregating DNA, so you have to replicate the DNA and you have to segregate it. So one molecule becomes two, and then you have to partition it into two daughter cells. And to do that, because these DNA molecules are very long, and they would intertangle with each other when you're pulling them apart, at one point in the process, you compact them into their most compact structure, these mitotic chromosomes, which you see in all the textbooks. They are often depicted as these sort of X-shaped molecules. So you've gone from 2 nanometers to 1,400 nanometers. Okay? You can actually see these in the electron microscope. So this is DNA attached, this is this structure here. And you can see the ends of a chromosome here, and the end is, one has come over the other like that. What you're looking at, each one of these blobs, is the top of a loop that's coming out from the bottom of the chromosome. So at this most condensed phase of cell proliferation, the chromosomes are all, the DNA is all looping out, wrapped into these sort of structures here and appearing out. Now, what happens if you take that and remove those histone proteins? This is what you see. So this is the residual protein scaffold, which still retains the shape of the chromosome. And now all the DNA, you can just about make out little strands of DNA. This is naked DNA, and it's just spewed out. And all because of that negative repulsion, you cannot compact it into that shape. Okay? These are remarkable photographs of chromosomes and the DNA that's in them. This is an old one. I remember seeing this in a textbook as an undergraduate back in the 1980s. Okay, so here's one of these chromosomes. This is at its most condensed state. Okay? This is what happens in cell proliferation from the moment of conception to the time we die. This happens about 10 to the 16 times. Okay? A single chromosome duplicates to form two sister chromatids that are then segregated into the two new daughter cells. So each one of these will be wrapped around a new cell membrane to form a new cell. So this happens, there are 10 to the 16 divisions in a typical human lifespan between the moment of fertilization and death. That's a lot of divisions, actually, for a lot of us. It's a remarkably accurate process, that you can have all those divisions and we can live to be 70 plus years without too much wrong, for a lot of us anyway. Okay, I just want to show a brief movie from one of my colleagues in the Center for Chromosome Biology, Kevin Sullivan. This is sort of work he does. He's very interested in that process where the chromosomes are pulled apart, the segregation process, into the two new daughter cells. And there's this rather, there's this rather beautiful structure called the mitotic spindle, which you saw in green. It'll cycle back in a minute. What that is it's a structure made up of cables. There it is there, on its own, the green stain. So there's two microtubule organizing centers here, and then each of these cables, or bundles of these cables, comes out and attaches to these red spots, which are the primary constriction in the chromosome. When you see those chromosomes, those X structures, you see in the middle there's a narrow area. That's what these spots are here. They're where the cables attach. Once you have cables attached either side of a chromosome, and all the chromosomes are attached, then what happens is the cells pull those chromosomes apart into two new daughter cells. Okay. So that's just a brief whiz through the history of genetics, what Mendel did, what genes are, and what chromosomes are. Okay. So this is the whole basis of genetic testing, DNA and chromosomes. So what should we, I want to talk a bit about genetic testing. Obviously, the analysis of DNA and chromosomes, which is your genotype, what we call genotype. But genetic testing can also be proteins, sugars, fats, because the consequence of your genotype is your phenotype. We just mentioned two genetic tests earlier on, the earlobes and the widow's peak. That's a genetic test. It's a very simple genetic test. Brown eyes, blue eyes. Every time you look into someone's eyes and say, you know, you've got beautiful blue eyes or beautiful brown eyes, you're doing a genetic test. Because eye color is, brown eyes are dominant over blue, okay? So if one parent has got two brown alleles and the other has two blue alleles, 3 out of 4 of those children will be born with brown eyes, okay? That's a genetic test. So genetic testing doesn't have to be sequencing DNA or anything like that. It can be just looking into someone's eyes. Okay, so this is what we mean by, well, some molecular phenotypes would be using protein, sugars, fats, metabolites to analyze phenotype. But the external appearance that we've just discussed is another type of genetic testing. Now, in medicine, we're using these genetic tests in order to detect mutations, including chromosome abnormalities, for example, in Down syndrome. That process I showed you in the movie a while ago, where the chromosomes are successfully pulled apart, is an incredibly accurate process, but just occasionally it doesn't work right. And one cell inherits 3 copies of chromosome 21, and that's all Down syndrome is. 3 normal copies. There's nothing wrong with any one of those copies. They're absolutely normal. It's just having 3 copies. It's the genetic imbalance of having 3 chromosome 21s rather than 2 that causes Down syndrome. So all those amazing phenotypic consequences of Down syndrome children are all caused by simply having an extra chromosome 21. So what do we use genetic testing for? Well, carrier screening for Mendelian diseases such as cystic fibrosis. Parents like to know if they're both carriers. If they're both carriers, and 1 in 20 people in Northwest Europe is a carrier, if they're both carriers, they have 1 in 4 of their children will be affected by cystic fibrosis. So you can do something about it if you know. Unfortunately, what that means is identifying a fetus that is affected and aborting it. And we can't do that in Ireland, of course, but it is an option in many parts of the world. Pre-implantation diagnosis, so same sort of thing. This is for IVF embryo screening. You want to screen the quality of the embryos because you want to have the best chance of that couple having a child. That's what they're there for in the IVF clinic. So you screen the embryos to make sure they are high-grade embryos. Prenatal diagnosis, we just talked about Down syndrome. That's another good example. Genetic genealogy, this is becoming really popular. There's money to be made in this area. People are interested in their maternal and paternal ancestry. We can do that because there's a part of the Y chromosome that determines maleness that doesn't recombine with the X chromosome. It always stays. It's like the male surname. It stays, it runs in the male lineage. So there's a part of the Y chromosome that identifies the male ancestors going back to the very beginning. Elsewhere in the cell, there's another organelle called the mitochondria that's inherited precisely, almost exclusively down the maternal lineage. So you can determine your maternal ancestry through looking at the mitochondria, and you can determine your paternal ancestry by looking at this region of the Y chromosome. You can, for a couple of $100, a couple of €100, you can determine your genetic genealogy, your ancestry. It's also used for pre-symptomatic testing for adult onset disorders. A very good example is cancer predisposition. Now, most cancer is not inherited by bad genes, by faulty genes, but there are some notable exceptions. In hereditary breast cancer, the BRCA1 and the BRCA2 mutations are particularly, for example, if you have a BRCA1 mutation, both alleles, it's a recessive trait, are defective in BRCA1. Females will have a very high chance, 60 to 80%, that they will develop breast cancer by the time they're about 40. Knowing this, some women choose to have mastectomies, so they can't get breast cancer. So that will save the lives of lots of women. Increasingly, diseases like Alzheimer's diseases we're beginning to understand the genetic basis, and there are now emerging genetic tests that address your susceptibility to developing Alzheimer's. Same with Huntington's disease. There are many, many examples. It's also used in diagnosis and prognosis, what condition a patient has and what it means for their the outcome of their disease, what chances they have to survive, for example, in cancer. Okay, these genetic tests are exploding. They're increasing exponentially. In crime, you've got forensic identity testing, crime scene, maternity cases in the legal profession, very easily addressed with modern genetic technology. And then there are research tests, where you do genetic testing for gene discovery purposes, how genes work. This is really the rate-limiting step in the whole genome project. This is going to be the hard thing. Sequencing human genomes is now easy. It's a piece of cake. Determining how genes work is the hard thing. Now, in these research tests, the data obtained is usually not available to patients or their doctors as anonymized. And a new area is pharmacogenomics. The influence of your genetic variation, what makes you you as an individual, on drug responses. For example, if you've got gene mutations, it's a loaded word. Mutation has negative consequences. If you've got a genetic variation that makes you susceptible to a drug, and if you don't know it, the doctor gives you the drug, it has serious consequences, including death. It would be nice to know what drugs to give to what patients based on their genetics. So this is a new area that is developing very well. So what's the procedure for genetic testing? Currently thousands of these tests available, and like I said, it's growing all the time. Well, informed consent is required for medical tests, and for medical tests, genetic counseling is also required. And it's performed in biological samples. Blood, buccal smear is very easy. You just put a swab inside and you just rub it against your cheek and send it to the lab. You need very little material. Hair, skin, amniotic fluid, or semen in crime scenes. It's sometimes difficult to interpret, and the type of test, the family and personal history also needs to be considered. So what about the results? Well, a positive test can confirm a risk of developing a disease. It can determine your carrier status, the diagnosis or prognosis for your disease, your biological parents, your identity, your ancestry. But there are always false positives. With any scientific test, always be aware of false positives. Negative tests can demonstrate the lack of a genetic variant linked to a disease, that you're not a carrier, so you don't need to worry with respect to cystic fibrosis. One partner might be a carrier. If the other isn't, there's no problem, at least for the next generation. Lack of a genetic relationship. False negatives are also positive, and cannot yet test for all possible genetic alterations. We're just really, we don't have a complete kit for testing for genetic alterations. A lot of tests are actually uninformative or ambiguous. They cannot confirm or rule out a disease risk or prognosis or genetic relationship. And you sometimes can't distinguish between natural variants and variants linked to disease. We are all mutants. There's no such thing as a wild-type human. Most of the mutations in our bodies, in our genomes, are silent. They have no phenotypic consequences. You have to be able to find the needle in the haystack between all that variation. So most of our variations have no impact on health. So results often require confirmation, and here's a nice example. I found a cartoon. So you don't always believe what you hear, okay? You need to confirm, okay? Right. Another recent phenomenon, particularly in America, as always, America leads the way. In westernized society, America is really the driving force, it seems to me. And these are direct-to-consumer genetic tests. They are genetic tests that the consumer can access directly over the internet. Just order a kit, take a buccal smear, send it back, and get the results without any intervention or discussion with a healthcare professional. They are available right now. Any of us could do this for medical conditions. For example, these BRCA1/2 mutations and cystic fibrosis. So anyone can do this independently of the healthcare profession. Benefits would be consumer accessibility, promotion of proactive healthcare, privacy of genetic information, okay? But the problems, there's a complete lack of regulation, exaggerated and inaccurate advertising, lack of professional guidance, therefore misinterpretation of results. People get these tests and they often get paranoid. Perhaps the people who want these tests are probably paranoid in nature. It's controversial. The scientific community opposes them. Here's an example I found on the web. You've got things like easydna.ie. I like this one, who'sthedaddy.com. Okay, so let's move on to the Human Genome Project. 23 pairs of these chromosomes. I showed you electron micrographs and fluorescent microscopy of these chromosomes. 46 chromosomes in a diploid genome, just over 6 billion base pairs in that diploid genome, plus that small mitochondrial DNA genome that doesn't live in the nucleus, it lives elsewhere in the cell. That has just 37 genes. This is the thing that's inherited down female lineages. The latest estimates are 20 to 23 proteins encoded. That's just 1.5% of the genome. So if you're a biochemist interested in proteins, the molecules you're interested in are encoded by 1.5% of the genome. But most of the rest of the genome is now known to be transcribed into RNA. This was a surprise to us in recent results. So the transcriptome is twice the size of the genome because there's two DNA strands, and both DNA strands can be transcribed into RNA molecules. And we just do not know the function of all that RNA transcription. Some of it, no doubt, is background noise, but already we know that some of it is very important indeed. There's an awful lot of mysteries in the human genome. It was sequenced in a competition between a public group funded by governments, particularly America, led by Jim Watson, the guy who discovered what Francis Crick the structure of DNA. They published their paper in Nature, the British journal Nature, on the 15th of February, and then they had some competition from the enterprise sector in America, which was published the next day in Science, so Nature won that particular competition by a day, which was led by Craig Venter. Now, this was a draft. It was only 90% complete. The genome, by the way, is currently only 92.3% complete. Everyone thinks the genome's done. It's not. The most difficult bits to do still remain to be done. And in fact, one of the difficult bits are for aficionados, the ribosomal DNA genes in the human genome. I have a colleague in the Center for Chromosome Biology who's actually sequencing that region at the moment, and he's got some nice data that he hopes to publish soon. So hopefully Galway will be contributing to finishing part of this incomplete, or this part of this missing sequence very soon. Genetic variation, what does that really mean? Because you hear it a lot. Well, if you have a variation at one of those bases, so instead of an A base, you have a G base. That's called a single nucleotide polymorphism, often abbreviated SNPs. You hear people talking about SNPs the whole time. All a SNP is a point in the genome where there's a single point variation in the genome. In a typical human being, there's one of these in every 1,300 base pairs. And what did I say we had? We had 6 million base pairs. So there's an awful lot of SNPs in the human genome. That's what makes us individuals, or some of those explain why we're individuals. There are repetitive sequences in our genomes, and these are highly, highly variable between individuals. And this is, in fact, the basis of genetic fingerprinting. So looking at these highly variable regions and seeing how long or how short they are, we can assign, we can identify suspects in crime scenes. We can do paternity testing, maternity testing. Then you have these copy number variations. In this case, it's not a single base change. There's a whole chunk of the DNA. It could be millions of base pairs, and it's either removed or it's duplicated. So there's more than one copy of it. Now, there are typically 100 of these in each one of us in this room, accounting for about 3 million base pairs of information. Okay? What I want to say about this variation, so there's an awful lot of variation in the human genome, but in some senses, there's very little. And that's because the human beings have come through an evolutionary bottleneck back in Africa. We are actually all incredibly related compared to other species. There's more genetic variation in slugs than there is in humans, okay? And that's because we went through an evolutionary bottleneck. This makes it good news for the genome because I said there's a lot of genetic variation, but relatively speaking, to other species, it's small. It's more manageable in that sense. So to date, we've identified, because of the Genome Project, nearly 3,000 genes underlying Mendelian diseases. Prior to the Human Genome Project, there was just less than 100. So that's an amazing, that's an exponential increase in identifying genes involved in Mendelian diseases. We also found just over 1,000 genes involved in common polygenic disorders. Things like susceptibility to cancer, susceptibility to heart disease, things like that. height is not a disorder, but it's exactly the same thing. It's a polygenic trait. But I have to say, I've got this quote from a recent review from Eric Landa, one of the leaders in the field in Nature, where he was looking at discussing the Human Genome Project 10 years on after its publication. And he makes a very good point. The greatest impact of genomics has been the ability to investigate biological phenomena in a comprehensive, unbiased, hypothesis-free manner. Okay? So that's still the greatest benefit. The practical benefits are still in the future, largely in the future. Now, I want to consider one human gene, and this was published, I think, in 2008, and it's Jim Watson's genome. So this study was really to demonstrate this new way of sequencing DNA. This is massively parallel DNA sequencing. This genome was done in 2 months, and it only cost, in this case, I think, yeah, it only cost $1 million in 2 months. This is 2008. Previously, it was 100 times that. Around the same time, Craig Venter published his genome, because he's always competing. They don't like each other. Jim Watson and Craig Venter are mortal enemies. I think Venter used to work for him, and they don't like each other. So they're always competing against one another, real alpha males. Venter did it the old-fashioned way. There are some advantages to the old-fashioned way. So Venter sequenced his own genome for 100 million. And these guys with this new technology sequenced it for 100th of that. Okay. So what do we know about Jim Watson's genome? Because it's instructive. Like I said, it was done by this next-generation sequencing method in just 2 months. It cost $1 million, 100th of the cost of traditional methods. What Jim has got in his genome is 3,300,000 SNPs, these single nucleotide polymorphisms, relative to the reference genome from the Human Genome Project. 82% of the SNPs were previously known. This suggests that we're beginning to identify most of the SNPs that exist, at least in European populations. Most of these SNPs are presumed to be neutral, as I mentioned, no phenotypic consequence. However, Jim has 11,000, and of those 11,000, 85% were previously known, resulting in altered proteins. So 11,000 of those SNPs are altering the proteins in his, what we call proteome, which means they could have phenotypic consequences. He has 23 copy number variations, ranging in size from 26,000 base pairs to 1.6 million base pairs. 9 gains and 14 losses. So he's got 9 copy number variations where he's duplicated or even more, triplicated those regions, and 14 where he's lost those regions altogether. Interesting, he's got 1 region of homozygous loss. So he's got 1 chunk, I can't remember what size it was, of his genome that he's just lost. It's gone. He's a total mutant in that area. He's a complete null mutant for that region of his genome. Maybe that explains some of his personality issues. I don't know. This is a very typical result for natural human variation. This could be any one of us up here. It would be the different variations, but this could be any one of us. Now, currently, it's extremely difficult to extract medically relevant inferences from individual genomes, including Jim's. In fact, we couldn't even make a rough prediction of Watson's height from his genome sequence. This is the problem. You know, we're running away with ourselves, getting overexcited, but until there's a lot more science to be done before we can really utilize the potential of the human genome. Now, I brought out this sample because I was aware of this. Watson has redacted part of his human genome. So even this great, leader in human genetics was a bit sensitive about part of his genome, and it corresponded to the region of the genome encoding the ApoE gene. Now, the ApoE gene is implicated in Alzheimer's, and his mother suffered from Alzheimer's, and one of his sons is bipolar. Okay, so he's very sensitive about that. Why? I don't know why he did that. I mean, he let everything else, but for this one gene, he got a bit sensitive and didn't want it published, so he took it out of the database. However, human geneticists are very cunning and clever people. They realize because variants are very close together, stay very close together, they're not recombined, they're not moved from each other, they could infer the genotype of his ApoE gene, even though it wasn't in the sequence, from surrounding genetic markers. So then he went back in and now removed 2 million base pairs either side of the gene. Very sensitive and surprisingly sensitive. I don't know why, but it's just an interesting, particularly for you guys interested in ethics, that Jim Watson himself should be sensitive about one region of his genome. He was quite prepared to bear it all to the world, bare his soul, until he saw this, and he pulled it out. He redacted it. So what are the future perspectives for genomics? Well, the 1,000 Genome Project, in fact, it's a lot more than 1,000 genomes. This project is designed, and it's only possible with this next generation sequencing, to identify most, if not all, of the genetic variation that exists across all humans. And what they're aiming for initially is variants that are present in the population at least 1% frequency, and in the protein coding genes at least 0.1%. There will be very, very rare variants around the world, and let's focus on the important stuff first. Well, that doesn't mean they're not important, but let's focus on the easy stuff first. Now, sequencing costs for human genome are falling very dramatically. This is defying even Moore's law. The sequencing costs are just plummeting like crazy, and it's due to come down to a $1,000 genome pretty soon. It will be possible very soon for every one of us in this room to sequence our genome. What we do with it, I don't know. Okay, it would be very useful scientifically. Some people are even talking about eventually coming down to $100 for human genome. In a sense, your human genome is it's the most rigorous ID card you have. At $100, you could sort of sequence someone's genome before you let them into your country. Find out who they really are. I think it could happen because the next generation sequencing truly blew my mind. This next generation sequencing technology, but there's a third generation coming along that will probably decrease cost down at least to this and maybe getting close to $100 genomes. Remarkable. So whole genome sequencing will be used in medicine for diagnostic evaluation of children with major intellectual disabilities, autism, birth defects, development delays of unknown cause. Diagnosis. People want to know what's diagnosis and prognosis, okay? Sequencing the genomes of patients will be used to assess their suitability for specific drugs. This is the pharmacogenetics. Or different treatments. I happen to know of mutations in a gene that we work on called ataxia telangiectasia mutated. These individuals are very sensitive to radiation. So if they get a tumor and you do radiotherapy on them, you can kill them, doses that wouldn't kill other people. So it's very important to know someone's genome before you start messing with them in a medical sense, okay? Comparing patients' genomes with their parents will identify newly arising mutations. The eugenics movement way back, this horrible discredited movement, wanted to purge the human genome of defective genes. You can't do it because they arise all the time. Mutations are a natural consequence of being human. Even if we got rid of all the mutations and what we consider harmful mutations to the genome, they will come back at some frequency because even though the process of duplicating the DNA and segregating it is so incredibly accurate, you always get mistakes, okay? Couples must sequence their genomes before having children. As I mentioned, if you're heterozygous for certain alleles, or you got a lot of genetic relationship with your partner, it's very likely that 1 in 4 children could have some disease consequence because of your identity. Now, we've known this in society primitive societies have known it for years. Consanguineous marriages between first cousins, all societies, primitive societies, have outlawed that because they know from example that when you see the consequence of this, it's due to people being very closely related, then you expressing mutations that if the people weren't as closely related, simply wouldn't happen. Okay, but we can do it in a modern way, not yet, but presumably by sequencing everyone's genome and not just knowing if they're carriers for cystic fibrosis, but they could be carriers for there will probably be there are thousands, tens of thousands of Mendelian traits. By sequencing your genome before you have children, you can actually see what the possibilities are. Of course, that requires another type of intervention. Cancer genomes will be sequenced to identify best treatment regimes. This is already happening. It's costing too much to be used for old parents. It's a research technique at the moment, but it will be, I think, quite soon applied to cancer patients in the genome, in hospitals. And this is an area that's really interesting. Reconstruction of some of the salient features of human history. Structure of the ancestral human population in Africa. With enough genomes, we can go back in time and reconstruct the genome of the ancestral human. Population dispersals throughout the world, you know. Are the Irish Celts? No, we're not. We're pre-Celtic. Celts are sort of Germanic, you know, Transalpine Gaul. It's Celtic culture that came to Ireland rather than the Celts themselves. And interestingly, there's actually gene flow with archaic hominids. What that means is we actually interbred with primitive cavemen. You know, Neanderthals. We Europeans and Asians, 4% of our genome comes from Neanderthals. There's not a trace of Neanderthal DNA in African populations. So the Africans are more related to Homo sapiens than Europeans are. We have primitive caveman DNA in our genomes. Africans don't. I love that. Okay. Right, a future landmark that I don't know when it's going to come will be not just 1,000 genomes, 1,000,000 genomes. And it will keep going. I think it's possible that one day, in the West anyway, all of us might decide, or it might be decided for us by the state, to sequence our genomes at birth. I don't know. What's the major problem? I'm just finishing up. This is my last slide. Genome sequences, that's all these functional elements that have disease consequences or functions in the body, are just the vocabulary. That's all it is, which is not much use without the grammar. By grammar, I mean the rules that govern how functional elements are integrated together to create us, individuals, you know. Obtaining the vocabulary is now really easy and getting easier at an extraordinary rate all the time. But working on the grammar will keep us busy for millennia. The primary goal of human genomics is to improve the treatment of disease through understanding the underlying molecular pathways. Okay? That's the primary goal of human genomics. The secondary goal is to provide patients with personalized risk prediction. And we can't stop this. This is like trying to hold back the tide. You saw those internet sites, you know. The commercial world will, where there's money to be made, the commercial world will offer a service, okay? I don't think we can stop it. We must regulate it. And a final note, I think genetic technologies will greatly benefit mankind, but will also be abused by some. That's true of everything. All technologies get abused by some. That's why regulation is important. But the regulation should be fully cognizant of the science. That I'll leave it. Thank you very much for your attention.